Okay. This is the first part of the nervous system unit. Out of all the organ systems for the MCAT, this is probably one of the better ones to really know because they tested a lot on biology, but there's also on the psych section, they can ask a lot of this stuff as well. Not so much this is more of a cellular perspective, but for the next unit, when we go into the anatomy and then the sensory organs, definitely testable for psychology as well. Any questions on the homework or anything before we start? Or if you just want to wait until we go over something, you can ask a question then. Okay. Here's an overview of the topics. So again, this is just the beginnings of the nervous system, building things up from the cellular level. So there's neuron structure. I did not write any words on this. We can go through each of these terms. Hopefully, a lot of this should be pretty familiar. So here's sort of your generalized neuron that you would see in the central nervous system. The most important things are the axon and the dendrites. We're going to see in a minute. The dendrites receive signals and the axons transmit signals. And the important thing to note here is that this cell has multiple dendrites, and most cells do have multiple dendrites, but there's only ever one axon. So, even though this branches here, these are not separate axons. There's only one axon ever. Cell body has the nucleus, just like any other cell. You'll hear that called the soma. The axon hillock is right at the beginning of the axon, between the cell body and the axon, and that is where action potentials begin, as we'll see. Some cells, some neurons, not all, but some will have these sheaths around little segments of the axon. The sheaths are made of a lipid called myelin, and they are laid down by cells called Schwann cells or oligodendrocytes, which we are going to see in a second. And the spaces that do not have myelin are called nodes of Ranvier. Here's what that would look like under a typical microscope. These cells are the neuroglia, which we will see in a minute. This is just an overview of some different structures. So your multipolar was this one, kind of your classic neuron. And the reason I show that is because this is by far the most common. Pretty much most or all of your neurons in the central nervous system are going to be like this guy. So many dendrites, one axon. Bipolar are very, very rare. Most common in the eye, the retina, and they're literally just called, you'll see with the sensory unit, they're literally just called bipolar cells in that case. Unipolar, which are also called pseudo-unipolar because in the embryo they start off as bipolar. These are also common in sensory organs. Other cells, not super important to memorize the details of each of these. This is just more to show you that the nervous system it's made of neurons, but it also is made of these other cells that collectively we call them glial cells, those little cells you saw back then. So these first three are for the central nervous system. Astrocytes, most importantly, you would think for the blood-brain barrier. Ependymal cells line the ventricles and produce the cerebrospinal fluid, which is sort of a lubricating and protecting fluid in the central nervous system, which we'll learn more about later. Microglia are essentially immune cells, and the equivalent to those in the peripheral nervous system are called satellite cells. Not sure if that's testable though. And then these two are more important. So Schwann cells and oligodendrocytes both produce this myelin that we saw here. The difference is Schwann cells are for the peripheral nervous system, and oligodendrocytes are the central nervous system. And oligodendrocytes actually look different because they can actually wrap around multiple cells at once. You'll notice each of these are actually one Schwann cell. Hold on, I think someone wanted to join, maybe. Oh, nope. Oh, I had this question on oligo versus Schwann cells on my full length today. Yeah, I don't know a good mnemonic for that, but it is something to know. Of course, they would never show you a picture and have you know the difference, but just for your knowledge, there are multiple oligodendrocytes myelinate multiple cells at once. Resting potential. So this is kind of our starting point for going into action potentials and the electrical transmission of signals in neurons. Number to memorize, resting potential is negative 70 millivolts, and it's negative. The inside of the cell is our reference point. So negative 70 millivolts means that the inside of the cell is more negative than the outside. Well, what makes it negative? Charged ions. So we have either more positive ions on the outside or more negative ions on the inside, or both. So the things that create this, when we're talking in the context of neurons, we're going to think there are a lot of ions in cells and outside of cells. We also have, chlorine, calcium, which we're going to see later as well. But the two big ones we think of here are sodium and potassium. Something to know is that there's a lot of sodium inside the cell and a lot of sodium outside the cell and a lot of potassium inside the cell. And the thing that maintains this resting potential is what's called, because, we want to move towards equilibrium, right? So if we had nothing around, we'd want to move this to have zero, right? Equal ion concentrations on each side. We use this pump here. So this is an active method of active transport. It's moving against the concentration gradient. It costs us 1 ATP. We take 3 sodium for 1 ATP. We take 3 sodium out of the cell and 2 potassium in. And that keeps sodium high out and potassium high inside. And notice what we're doing. We moved 3 positive ions out and brought 2 in. So we're removing positive charge from the inside of the cell each time we run this. So that's contributing to this negative 70 millivolts. However, actually making it even further, so the key here is that potassium is actually the major ion contributing to this. There's some math you can do to show that. It's not really important here, but potassium is what's contributing to this, and part of that is these non-gated potassium channels. So passive transport, they're always open, and we're bringing in potassium. So potassium has a higher concentration inside the cell. We have these channels here that are allowing it to flow out, which is taking positive charge out of the inside of the cell and making it more negative still. So action potentials are that's the term for electrical transmission of signals within a neuron. We're going to see, obviously, the purpose of the nervous system is to transmit information. That information is transmitted electrically within the cell and chemically between cells. We're going to see between cells in a minute. So action potentials. So the first step of this is that some sort of impulse changes ion concentrations and changes the potential of the membrane, right? If we can make the membrane more positive or more negative. How does that happen? We'll see at the very end. So it's a cycle, right? So we're starting kind of just at a point here. And there are 2 terms to know here. A cell can depolarize, which means that the membrane potential is higher, or it can hyperpolarize, which means that it decreases. So depolarize means it's more positive. hyperpolarized means it's more negative. And that's relative to that negative 70 millivolts from before. So if the depolarization isn't enough, and we'll see later why it might not be enough, if it's not enough, nothing happens. But if it goes to a certain number that depends on the cell, but it's usually about negative 55 millivolts, and for the same cell, it's always the same number over time. If we can raise that potential to negative 55 millivolts, we generate an action potential. And something to know here, this will come up with muscles as well. It's called the all or none law, or the all or nothing rule. So axons, or action potentials, it's information moving down the cell, an electrical signal. How strong that is and how fast it is set by the cell itself. It's set by the myelination and thickness of the neuron. It has nothing to do with how strong the impulse is. So for a cell where the threshold is what this is called, where the threshold is negative 55, doesn't matter if we go up to way above this, doesn't change anything. We generate the same action potential as if we had brought it to this exactly. So here is a picture, kind of a classic photo of this, which goes over all the steps here. So you'll see here, and some of these numbers are more important than others. So there's the negative 70 we talked about. So this flat part here, we started at negative 70 millivolts. Something happened. We'll see what that something is in a minute. That this first hump here that raised to about 55. And once it hit 55, we notice we get a sudden depolarization that is much larger than the initial one. This is caused by voltage-gated sodium channels. So remember, there was a lot of sodium outside the cell. These are passive transport, but they're normally closed. When we go to about negative 55 millivolts or above, these all open, and all that sodium outside can rush in. Well, sodium is a positive ion, so it's going to raise the potential a lot. Now, at the same time, we also have voltage-gated potassium channels in the cell. And these open slower and take longer to close. So at this apex here, the sodium channels are closed and the potassium channels open at this tip here. But it's not, we don't call it closed. So down here, we say they're closed. During this point, we say they're inactivated. So they are closed, but it's a different state from here. So ions can no longer flow through them. So we've blocked them and we've opened up potassium channels. Well, potassium is high inside the cell. So positive ions are coming out of the cell, and so that rapidly drops the potential. And it actually drops so quickly that it overshoots the initial potential here. Notice it dips down below negative 70. That's called hyperpolarization that we talked about before. And remember, the sodium-potassium pump is acting to maintain that when it is acting, then the equilibrium is now negative 70. So its action then brings it back to negative 70. So with this, the refractory period. So we said before here that a strong signal can affect how fast it fires. Well, there is a hard limit to how fast a neuron can fire. And this is caused by the absolute refractory period is caused by these sodium channels becoming inactivated. So remember, at the apex here, those sodium channels are inactivated. And they do not go back to being closed. Remember, they had to be closed before they could open. They do not go back to being closed, and so we can't open them again until they go to the resting potential. So remember, sodium opened after this impulse here. The sodium channels opened. At the apex, they're inactivated, and they're done. They cannot respond to a depolarization again until they get close to the resting potential and are back to the closed state. So at this point, you can never generate a new action potential no matter how strong of an impulse you do. Now, the relative refractory period is longer, and it's caused by the hyperpolarization. So notice for a little period after here, you could notice for this period here, the resting potential was lower. So we're going to see with summation in a minute that it takes a stronger impulse to raise it. There's more of a difference to make up to get to negative 55 now, right? So it now takes a stronger impulse, and that's the relative refractory period. But of course, eventually it will return to the resting potential, and then it's just like before we started. So that's action potentials. Now what happens then? Well, there's a good photo in the Kaplan book that I forgot to include here, but basically we start this process somewhere near the cell body in the axon hillock, the beginning of the axon. Well, the cell membrane is becoming depolarized. The inside of the cell is becoming depolarized. We're saying that the membrane is becoming depolarized. That can spread to nearby areas. So when this is happening, when this depolarizes, we get this little hump here, that's localized somewhere, right? Well, then we get this sudden depolarization caused by the sodium. This positive charge spreads to nearby areas, which then causes those voltage-gated sodium channels to open, and we do this again over there. And it keeps going down and down and down. But this refractory period means that we can never go backwards. So we talk about axons and dendrites. The key here is that we're always going from dendrite to axon. So we're going from the part of the axon that's closest to the cell body away from the cell. You can't go backwards. Now, in myelinated axons, the point of the myelin is a lipid, so it prevents ion movement. It's hydrophobic. So in a myelinated axon, if you were able to see, if you were able to zoom in and see the charge moving, in this wave-like pattern, it doesn't move through the myelin. It jumps across the nodes of Ranvier. So those are the unmyelinated parts. You would see this wave of positive charge jumping between the nodes, and that's called saltatory conduction, and it's much faster than unmyelinated axons. So in cases where speed of transmission really matters, we will have myelinated axons. So that's electrical transmission. Now what happens next? So now we've done this wave. Let me check if there are any questions. Nope. Okay. So we have this wave here. Now we're at the end of the axon. What happens next? So now we're going to transmit this to the next neuron, and this is called this is chemical transmission at this point, and it occurs across a synapse. A synapse just means like a small space. So the key here is that neurons are not connected. There's actually a very tiny space between them, and it's always between axons and dendrites. And we talk about a presynaptic and a postsynaptic neuron. So the presynaptic neuron has the axon, which communicates with the postsynaptic neuron dendrite or dendrites. But they are not directly connected. They communicate through the release of neurotransmitters. So let's just walk through this. So the action potential reached the end of the terminal. We had that wave of positive charge that hit there. Okay, positive charge. At the very end of the axon, there are voltage-gated calcium channels. Calcium A good fact to know, calcium is much more common outside the cell than inside the cell. Calcium has a few specific purposes in cells, so we keep it. There is very little calcium in the cell unless it's during a little moment like this when we're using it. So what happens is these calcium channels open and calcium ions flow into the cell at the end. Well, through a pathway that's kind of out of the scope of the MCAT, this then leads to exocytosis of neurotransmitters. So we've already been making these neurotransmitters, which are they're usually small molecules. They're mostly, there are a couple, that can be larger, but usually they're not. Don't think like big proteins with complicated structures. They're usually very small organic molecules. Sometimes they're amino acids, so just one amino acid with a few changes. And these have already been made, and they've been packaged into vesicles. And we get the signal. They get spit out at the axon terminals. So now they're floating around in the outside of the cells in this synapse in between. Well, the postsynaptic cell has receptors on the membrane. Usually the neurotransmitter does not diffuse in. Usually there are receptors outside. And this is key because neurotransmitters can do different things depending on where they're at. You'll see later on when we talk about specific ones, they have different purposes depending on where they are. Typically, the function of a neurotransmitter depends on these receptors. So one cell will usually only have one type of receptor for a neurotransmitter, but that determines how it responds to it. So different parts of the body, the same neurotransmitter does different things. And these bind, and where it binds, it might be an ion channel, or it might be a signaling pathway. Somehow downstream, it changes the membrane potential. And usually that's by opening ion channels. So these are, we might call the neurotransmitter a ligand. These are ligand-gated channels. So we open up a channel, we cause ions to flow, and that's going to change the membrane potential somehow. And we'll see why that matters in a minute. And of course, if we just left the neurotransmitter there, it would have that response forever. That's actually how some poisons and toxins work. So we get rid of them usually very quickly, and the two major things are reuptake. So essentially, there's endocytosis, and the presynaptic cell picks back up the neurotransmitter and recycles it, or breakdown. We can release certain enzymes like acetylcholinesterase or acetylcholine that very quickly breaks down the neurotransmitter. So, okay, the neurotransmitter has bound to the cell, the postsynaptic cell, and we've let in ions somehow. So what happens next? Okay, well, there are two categories. So we can have an excitatory postsynaptic potential, and this is whenever somehow through some exchange of ions via those channels, we have depolarized the cell. So that makes an active potential more likely. Remember, we got to hit that threshold of negative 55 millivolts. So in some cases, if we want, back at the beginning where I said, wait, and I'll show you, in a minute, what caused that initial impulse, this is what caused it. So if it's strong enough, which usually means if we have enough of these at once, or if it's a particularly important neuron, then we will hit that negative 55 and we will cause an active potential. And in this way, we're now conducting the signal down down a path of neurons, right? So we start somewhere with a sensory organ, and you'll see when you go over specific senses how those are ultimately also based on depolarizations. And ultimately, we're passing it from axon to dendrite to axon of multiple connected neurons. Now, inhibitory is the opposite. So somehow we've hyperpolarized the postsynaptic cell. We've made it more negative, which means that if something else tries to come in and depolarize the cell, there's now more of a hurdle to overcome, and it's going to be harder to make another action potential. So that brings us to summation. So many areas, many neurons. It's not where we said that there's multiple dendrites and one axon, but the axons can split. A lot of postsynaptic neurons, they have multiple neurons, presynaptic neurons, that are bringing signals in, and we call that integrating. Not all. So it's most common in the central nervous system, which is why we had those kind of classic looking neurons with many dendrites and one axon. In places like the muscle, each muscle fiber actually is only controlled by one neuron. But in the central nervous system, the purpose of the central nervous system, it's integration. So we bring things in from multiple neurons. And we're basically summing up the effects of them. And there's two things we can do here. So there's temporal summation. Remember I said before that the speed of a signal matters. So if we bring in a bunch of action potentials very quickly, basically if the presynaptic cell has action potentials that are as fast as the refractory period will let us do, well, we're now releasing neurotransmitters, and let's say it's excitatory. We're depolarizing that cell so fast, so many times in a row, that the effects are adding up. And even if one might not have been enough to reach the threshold, now it is likely they will be. Now, in spatial summation would be, okay, we have multiple neurons, multiple axons or multiple neurons that are releasing neurotransmitters for one postsynaptic neuron. And if they do that at very close to the same time, those effects are going to add. So you just say, okay, well, each excitatory input is going to depolarize it more. Each inhibitory one is going to make it hyperpolarized more. The stronger one wins. Okay, now we have practice questions. So are there any questions for this? First of all, I'm going to put it up so I can see the chat here. You all can still see the slide, right? Someone?
Yep.
Okay. Then we'll go through a few practice questions. Don't just take a couple minutes, ask whatever questions. Don't be afraid if you're wrong. So if a neurotransmitter binds to that postsynaptic neuron and we have an influx of chloride ions, would that be excitatory or inhibitory? Have an inhibit. Yes, because chloride is a negative ion. And this could happen. So chloride is more common outside of the cell. A good way to remember that is sodium chloride, like salt, right? Hopefully we kind of drilled it in that sodium is very common outside of the cell. So chloride is also common outside of the cell. And we open up a chloride channel, it will inhibit that neuron. Which of these ions directly controls the release of neurotransmitters from those vesicles? Have a calcium. Anyone else? Well, you're correct. Just a straight memorization question there. So this is after the action. So sodium and potassium are the ones controlling. It's all about influx of sodium and then potassium leaving. That creates that action potential graph. But once it reaches the end, it's this calcium influx that is signaling to release the neurotransmitters. There are multiple answers for this one. So which of these factors would increase the speed of action potential conduction? I have a B. Anyone else? Remember, there are multiple answers. Myelination. Let's see. So a thicker axon, that wasn't in the slides, but I briefly mentioned it. So yes, a thicker axon is faster. More frequent stimulation. So no, that would increase the frequency of action potentials, but each action potential would not move any different. The speed would not be any different because remember, the speed of the action potential is set in stone by the structure of the cell. Nothing about the impulse you do, about the magnitude or the speed of that impulse can change the speed of the action potential. But again, myelination, that's also structure of it. So myelination, we've now forced the action potential to jump across those nodes of RON-VA, which makes it faster. And again, increased magnitude of the stimulation. No, if you leave that depolarization there for a long time, you might have a whole bunch of action potentials, very quick, frequently. But again, the actual magnitude or speed of those action potentials never changes. Part of the neuron located at the synapse, the postsynaptic neuron. Which part is at the synapse? Which part synapses with the presynaptic neuron? Dendrite. Anyone else? Dendrite. Yep. Always goes axon to dendrite. Axon terminal, it goes down the axon to the axon terminal. Synapse, dendrite. If we made the sodium-potassium pump faster, theoretically, which of these would become shorter? Kind of relative. Okay. Yep, you're right. Oh, someone wants in. So remember that the absolute refractory period, that goes from that apex down to the resting potential. And the return there is caused by the influx from the voltage gated potassium channels. So the sodium potassium pump does not really significantly affect that. But the relative refractory period, yes, you're right, because we said that the voltage gated potassium channels overshoot the resting potential, and it's the sodium potassium pump that then restores the resting potential. Okay, so absolute period is like fixed. Yes, it's based off of the inactivation of the voltage gated sodium channels. Which you have to bring them back to the resting potential. And you have to bring them back to being close to the resting potential to then let them be opened again. And what's doing that is the potassium channels. So there would be a mechanism. We generally say, yes, you can't change it. So if we, again, another hypothetical here, if we could somehow take a neuron and raise the resting potential less negative, would action potential would occur have a true. Okay, sorry about that. Am I back, by the way? Someone pull the slides back up. Okay, we're good. Great. Okay. Sorry about that. So, yes, maybe I got cut off here. So, yes, if we've lowered the gap, we've lowered the difference we have to make up between negative. Remember, normally got to go from -70 to -55. So if we're closer to that -55, then we're good. We're going to be easier to make the action potential. Another theoretical question here: if I took the voltage gated potassium channels and just closed them, and they could never open, would a neuron ever return to the resting potential? Maybe a tricky one here. Take a guess. False. Anyone else? False. Yep, it's false. Never is a scary word. Yes, that's a good thing to know if you biology as well, and MCAT and ours too on the MCAT. The absolutes are usually not correct. There's usually an exception. So yes. So if we go back to that graph, this is my last question. Yes. Okay. So let me show you that graph again. And the potassium channel's opening is what dropped it, may cause this huge drop here. But remember, when the sodium potassium channel is operating, it wants to bring the cell towards -70. So even if we closed off, even if we didn't have the potassium channels to make this huge drop here, we would still have the sodium potassium pump that could very slowly bring it down back to the resting potential. So with that is the last of those. So we can go through the practice question here. Practice passage, sorry. Let me just give you the link to that. I'll stop sharing now. So take, 10 minutes or so on this, and then we'll go through the answers. If you want to let me know when you're finished too, we can get a gauge of where everyone is on this. And you have any questions, you can ask. Okay, I've got one done. We can wait. We can wait a little longer if everyone else is still working. Good to go. No, we can wait till 8:50 or a little bit later. Which question are you on? 3? We can wait. All right, let me share my answers then. Can you see the slide again?
Yeah, we can see it.
Okay. So number one, scientists opened potassium ion channels in the dendrite. What's going to happen? Well, potassium, we said, is more common inside the cell. So if we open the potassium channel, the potassium is going to flow out. So outward potassium flow would be the first thing where you could cross some off. Remember, elimination is usually the best strategy with a lot of MCAT questions. And then, okay, what does that do? Well, we've let positive charge out. So that's going to it's going to hyperpolarize. So we've made it harder to make an action potential in that case. This one's just a memorization question. You really should know all these ions, where they're more common. You don't need to know numbers. Please don't memorize numbers. But you should know where they're more common. But definitely these two you probably knew. So sodium is more common outside and potassium is more common inside. But then for your knowledge, chloride, you can remember that as sodium chloride going together. And then calcium, because calcium has such a crucial role inside the cell, we only let it in for things like muscle contraction and neurotransmitter release where it's really needed. Sodium-potassium pump is inhibited. They fire a train of action potentials, which stops spontaneously, and then the neurons cannot be stimulated into firing anymore. So we can eliminate a lot of things here as well. So, well, first, when they say likely explanation for this, that means they want to explain both of them. Well, hyperpolarization would not explain firing spontaneous action potentials quickly. You can never get an action potential you're never going to get an action potential by going below negative 70 millivolts. Decreased intracellular sodium, well, that's the same thing as hyperpolarization. So if two answers say the same thing, they both have to be incorrect. Increase intracellular potassium? Well, maybe, but that's just the normal action of the sodium-potassium pump, right? But depolarization can explain both of these because remember, we have to depolarize the cell, not very much, just negative 70 and negative 55. We depolarize it to cause an action potential. But then when we go much higher than that, remember, once we hit that apex, then the sodium channels, voltage-gated sodium channels have become inactivated, and they can no longer fire another action potential. So what's happening here is that we've stopped. The sodium-potassium pump is the thing keeping it, normal. Everything wants to be, zero everywhere, no charges. So the sodium-potassium pump is what's keeping it negative. So if we get rid of that, it's going to bring it closer to zero, that's depolarization. So we're going to fire an action potential, but then there's nothing to restore it back down because remember, this is what's doing that. So there's nothing to do that again. So it's just going to stay depolarized and those sodium channels are going to stay inactive. Okay, the scientist transiently opens sodium channels on the dendrite, which the following describes what happens. Well, there's more sodium on the outside. So if we open the sodium channel, we're going to make it positive and we're going to depolarize. And that's something that some excitatory neurotransmitters will do that. And then the last question, which conclusion about the type and direction of ion flow is most likely based on the results? So you actually only need one of the results because you can cross things off here. So if we go over to this, the results were that when the channel was open, the membrane depolarized. It went from negative 60 to negative 50. So let's ignore the other ones. The correct answer will explain these somewhat, but we don't even need them. Let's look at the wrong answers. Well, outward sodium flow, well, that wouldn't depolarize. Well, first of all, it never happens. You have to pump sodium out, but pumping sodium out actually hyperpolarizes it. Outward calcium ion flow, again, would never happen because it's more common outside the cell, but even if it did, we're pushing out positive stuff, so that's not depolarizing. Inward chloride ion, that can happen, does happen, but we're making the inside more negative, so we're hyperpolarizing it. So only this one actually depolarizes it. Okay, are there any questions about that? Please stay on. I have a couple of announcements, but does anyone have any questions on this?
Hi, yeah, I have a question. So can you please just clarify like calcium's role in the action potential? Like, does it just follow after the depolarization and repolarization phases?
So calcium does not really have a role. Well, I mean, this is tough. So we're talking about action, but just, I don't want to tell you stuff that's going to mess you up for later. Calcium actually can have a role in the action potentials of muscles, right? Have we covered muscles yet? I don't know. This is my first biology unit.
Yeah, I just know that like calcium binds with like troponin or something.
Yeah, it binds with troponin, but it's also that calcium flow in a muscle. It's not just about that. It's calcium has a role in depolarizing the cell, at least in heart muscles. The extracellular calcium there's actually extracellular calcium that can flow into the muscle and has a role in the depolarization, which again is not really the same as this action potential. For the purposes of neurons, the calcium does not actually have a role in the action potential itself. So where this graph I showed, let me go back. This guy right here, which is happening, remember, it's like a wave-like pattern. This is moving down and down the axon, right? This is strictly shuffling around sodium and potassium. However, once you reach the end of the axon, there are voltage-gated calcium channels there. So we've depolarized the end. We've gone all the way down to the end of the axon. We've depolarized. What happens then? We open up voltage-gated calcium channels. Calcium is more common on the outside, so the calcium flows in, and there is a signaling pathway that causes where that calcium then causes exocytosis of the neurotransmitters. So calcium's role is to be the transition from the electrical transmission of the action potential into the chemical transmission between neurons in the synapse. Does that make sense?
Yeah, thank you.
Anyone else? Okay, well, please don't leave just yet. If you've gone to the other sessions this week, hopefully they've shown you this, but this is the midterm of the course. Please fill out that survey. It's just for you all, and I do not look at the results anyways, but that will help the program as a whole improve, and they will enter you into a raffle if you do that. And of course, we do have the office hours. I have the office hours this Friday at 8 p.m. So if you have any more questions, or if you want to let this sink in a little bit more, we can talk then. And if you like what we're doing, then you can donate with our donation link there. All right. If that's all, we can end here. Thank you, everyone.
Thank you.